turn off my camera. Make sure I hit the camera button, not the leave meeting button. And this is the flyer that I had posted on our LinkedIn page a couple of days ago. Thanks everybody for joining today. And let me get on to the presentation. Um, so if you do have any questions after the meeting that we don't address, you can certainly download this Bioinformatics 101 primer that Afshin and Katie helped me write. This is the QR code for that. I just want to remind everybody that Katie and Afshin are people, not computers, so they may not remember things like the formulas that are underneath the the denominator for a t-test or something like that. So if you do have more detailed questions, the nice thing about this particular paper that I wrote is I peppered it with lots of references. So there's not a lot of math in it, but if you're curious about linear regression models, there are plenty of references about that and moderated t-tests and so forth. So you can look up directly the source material using that. And I'll put this back up again in a little bit. So what we're going to do is I'm going to give you a brief overview of Syngenics. We're hosting the meeting today. I thought it'd be nice for you to get to know us a little bit, and then we'll get right into the questions. So I just want to make sure I have an eye on time. So a brief presentation by me, and then we'll get into the questions. So Syngenics is a company that spun out of the University of Cambridge in 2008. Our chief scientific officer, Jonathan Blackburn, had some ideas, some biochemists regarding proteins and ways to make protein microarrays more effective, basically make them higher specificity, better reproducibility. I'll show you a little bit of the history and the technology in a minute, but he developed this technology in 2008. And in 2016, we opened up a facility in Malaysia to do the printing of the arrays. The company, Syngenics, we manufacture a protein microarray chip called the IOM, which has over 1,600 different proteins on it. It's a functional protein microarray that has been used in a variety of disciplines, especially in cancer, neurodegenerative research, and autoimmune research. It's very good at doing things like biomarker discovery, identifying patient endotypes or subtypes, among other things. And we'll see that as it comes out through the presentation. In 2020, Summa Equity became a majority shareholder for us, helping to develop our technology further, and we continue to develop protocols, core technology, and patents. The technology currently has 14 patents on it in different countries. And of course, we've partnered with a lot of different institutes throughout the world, including GlaxoSmithKline, as well as many universities like University of Birmingham, University of Leeds, the Mayo Clinic, NYU is not on here, but on NYU as well. All right, so what brings us here today? Well, I don't know if you know this. This was particularly interesting to me. When I started, my background is developmental neurobiology, and I spent 14 years with Agilent doing cellular respiration. So one of the first projects that I was interested in is, well, what are antibodies and autoantibodies, and where did they come from? So I don't know if you know, but antibodies were discovered in 1890. And this is going to be important because one of the core features of our technology, it's a protein microarray, right? And it relies on patient sera, which contains antibodies. So in 1890, antibodies were discovered. But I don't know if you knew this, autoimmune diseases weren't considered a category until 1964. Now, this is kind of important because if you take a look at the chart here, autoimmune diseases are categorized in 1964. Autoantibodies were discovered in 1957. It turns out autoantibodies are fantastic biomarkers. They can actually identify all different kinds of diseases. A lot of work being done on Alzheimer's and Parkinson's, and in our labs, we're working on cancer and COVID. There's a limited number of autoantigens, and these can actually reveal a number of different pathways to us that are involved in disease pathogenesis, the ability to predict outcomes for different patients on different therapies, to stratify patients into different categories. So these antibodies, these autoantibodies, wind up being huge for research and probably underappreciated. In 1965, in fact, was the first time autoantibodies were identified in cancer. I think it was breast cancer back then. Of course, the techniques to study autoantibodies were pretty rudimentary. Most of the time, you're just using immunohistochemistry. In 1971, ELISA was developed. So now we can put some protein down on a plate and then run some antibodies across it and illuminate them to find out, what kind of autoantigens might be there to help us with biomarker discovery and disease detection. In 1972, that's when bioinformatics was first coined. The nice thing about ELISA is it was able to give you somewhat of a high throughput when you're doing these types of experiments. There wasn't a lot of high throughput prior to 1970. Bioinformatics basically at the time described the communication networks that are occurring within and between not only cells, but also heredity. And this wound up running in parallel with biochemistry. So in 1972, bioinformatics is coined. It has a lot to do with biochemistry, and people worked on bioinformatics for decades. But it had a huge resurgence in 1995, and that's when DNA microarrays started to emerge. Now you've got really high throughput. And for me, this was like really eye-opening because I was a graduate student at University of Connecticut in 1995, and somebody came to the institute and gave us a lecture about this new technology called DNA microarrays. And he pulled a slide out of his pocket, and I was just like, "That's crazy." I mean, that's just like that's just heresy. That's how do you do that? How does that work? It's such a tiny amount of DNA. And remember, this was also the time the Human Genome Project got started. So there wasn't a ton. I mean, there was a good amount known, but we didn't have the genome mapped. So these DNA microarrays come out. I'm at a meeting, Society for Neuroscience, and a friend of mine is now involved in working with DNA microarrays. They're trying to identify differences in patient populations by running these assays. And she's showing me some of the data, and I'm like, well, how do you tell if something's significant? I mean, can you run a t-test on that? And she just laughed at me because my background was all about t-tests and ANOVAs. And 1995 was sort of a beginning of a new era of statistics. Now, you can see some things in the middle here. Recombinant DNA comes out in 1974. That's important because it gives us the ability to actually make the proteins that we might want to print onto a slide. And analytical protein microarrays actually predate DNA microarrays. In 1983, a fellow at a company near me in Exton, Pennsylvania, actually came up with a technique where he would spot bits and pieces of actually it was antibodies. He would no, it was cells. He put cells on a plate and then covered them with antibodies. A fellow named Sin, and that was in 1983. Analytical protein microarrays. Something else that was interesting. Now we get back to autoantibodies. 1986, the Finnish health study completed. That was a 10-year study and in of health and disease in the population of Finland. And one of the things that was interesting about the study is they found that rheumatoid factor, the autoantibody or the autoantibodies that were responsible for rheumatoid arthritis, actually predate the disease by up to 10 years. Right. So remember back in 1965 when they found autoantibodies in cancer? Well, now the idea is that perhaps autoantibodies could actually detect disease much earlier than any other mechanism out there. We've got the DNA microarrays in 1995, and then functional protein microarrays come out in 2002. So now we can look at the genome and the proteins that might be associated with disease, identify DNA mutations, variations, look to see if that's recapitulated in the proteome. And that can help us better refine our diagnostic tools as well as improve our ability to identify patients that might respond good to therapies. And in 2009 was one of the first times, maybe a little earlier than that, but that was the first paper I could find, where a protein microarray was used to identify cancer in a sample of patients. And then in 2018 was one of the first times that machine learning was applied to translational medicine. Now, machine learning is important because remember I told you the story about Society for Neuroscience? Like, can you run a t-test on that data? Well, you're talking about running experiments with hundreds of patients, multiple cohorts, and thousands of different features, whether it's protein or DNA. So you're going to need some help to go through that data and look for patterns. And that's where the machine learning comes in. And just so you know, I mentioned biomarkers are a big part of this. Well, the global biomarker market is expected to be $97.5 billion. So it's something that people are very much interested in. You're probably here partly because of that. And for me, biomarkers wasn't something we spent a lot of time talking about in the late 90s, early 2000s. If you take a look at PubMed, biomarker discovery as a field exploded in the last 10 to 15 years. So with Syngenics, our technology is a protein microarray, and we have a particular type of technique called CREX to produce correctly folded full-length proteins. The way this technique works is we code in frame a biotin carboxyl carrier molecule that's biotinylated. That's right here. So when a protein is produced and it's properly formed with its tertiary structure, the BCCP molecule is also properly formed and the protein can stick in the same orientation every single time onto the slide. So we know we've got a fully functional, fully folded protein. What's important about that is the folding, the shape of the actual protein matters when you're using antibodies. And the sample we use for the protein microarrays is serum from patients replete rather with tons of antibodies that we're going to wash across these slides and then look to see what sticks using indirect immunofluorescence. If a protein isn't properly folded, then you might get non-specific binding or no binding at all. So your false discovery rate is going to increase, your reproducibility is going to decrease, and of course, you may get non-specific results as well. So a misfolded protein, the BCCP molecule also misfolds, and it can't stick to the slide. So that's the idea behind the CREX technology that is at the core of Syngenics. You can see the patents down here for that. So I'm going to skip that. And I'm going to get into the questions now. But I just want you to know if you do have any questions for us, you can reach out to us on syngenics.com. You can follow us on LinkedIn. And then these are the business development directors throughout the world. In the Americas, it's Brad Nawa, and here's his contact info, and Andy Jackson, there's his contact info. And in Europe, you've got Adrian Churchman and Elvira Kaminska. And in the Middle East and Asia Pacific is Bermet Abalova. If you have any questions, you can reach out to one of them, myself, or contact us at syngenics.com. So let me go back over and see if anybody has a question or comment yet. Nope. All right. So Katie, let me get the questions I have. Oh, there it is. Okay. So first, I just wanted you guys to introduce yourselves, starting with Katie. What's your background like? How did you come to be here at Syngenics as the head of our bioinformatics team?
Thanks, Kevin. So I actually started out as a wet lab scientist. And as part of my PhD, I had cancer genomics that I was looking at. So I had gene expression microarray data, and I also had whole genome methylation data. At the time, there wasn't really anyone to do the analysis for me, and I was also interested in learning. So it was really out of necessity that I came to bioinformatics. Subsequently realized that I enjoy it a lot more than wet lab work, and I'm probably also a little bit better at it than wet lab work. So I think it worked out well. After my PhD, I did a postdoc in bioinformatics, staying with so my cancer research project for my PhD involved some microbiome work, which I was really interested in. So that was the focus of my postdoc work, was building pipelines for microbiome data. Following that, I supported various labs at UCT in data analysis. So that was really focusing on building pipelines for various data types, including microbiome, RNA-seq, metabolomics, and so also assisting with training postdoc students, postgrad students, so they could do their own data analysis. And during that time, I started working part-time for Syngenics to help them develop their pipeline. And I quickly realized that that was a challenge that I'd prefer to take on full-time. I was really interested in the technology. And so here I am. I've been there ever since.
Thanks. How about you, Afshin? How did you get involved in bioinformatics, and what are you currently up to?
Yeah, thanks, Kevin. Yeah, I mean, probably was similar. I think a lot of us from the older generation has a similar story like Katie's. So, my background is actually in physics. So I got my PhD in physics a while ago, and physicists tend to be a jack of all trades, but not an expert in everything. But you won't admit that, usually. Yeah. So I switched over to biology for my PhD in physics. And I was working on microbiomes first, and then I joined this cancer systems biology lab for the second postdoc I was doing. And I'm an academics, basically, and things like that. So, but I was doing a lot more wet lab work, just like Katie does. And then when microarray technology, as you mentioned, Kevin, started rolling out, people wanted to analyze it, and me as a physicist still had some computationally inclination. So, you know, me and some fellow lab members started teaching ourselves bioinformatics. There's no classes on bioinformatics when we started, so we had to come up with our own things and look at literature, call people up, hey, how do you do this? There was none of those nice forums that are now there, like BioStar and all these other ones that we all use. But you know, that's how it eventually started, because you know, the great thing is, you know, when you do the classical biology, you're starting like looking at one factor, one protein or one gene, and then expanding up there. But you miss the bigger picture of me being a systems biologist. You know, you want to see the top-down approach, see what's happening, and then I whittled that back down. So when bioinformatics first started, or omics and all the microarray technology, that was the big data that you could actually now start seeing the entire picture nicely. So that's how I got involved. I started doing a lot more computation. I still do both now, and then currently I have a couple of different projects I work on. From, you know, I'm at Blue Marble Space Institute of Science. I'm also at, you know, visiting research appointment abroad. I also have a nonprofit called COVID-19 International Research Team, which I lead. So I do a lot of different projects, from space biology work to COVID-19 to cancer biology, and then a bunch of other projects with other folks. And the great thing about bioinformatics is, once you have sort of a pipeline or an idea, you can do a plug and play method for a lot of different diseases. So you get, it's fun. Then you can actually not be stuck in working on one thing. You can work on 10 different things if you want, and still apply, you know, your knowledge and your pipelines to maybe sell things, you know, in different areas and different fields. But yeah, in a nutshell, that's what I do. That's who I am, and that's how I got involved with bioinformatics, because I think bioinformatics is fun, because now there's a lot of free data out there, you know, like publicly available databases, from, you know, cancer to space biology to lots of publicly available data. So even if you don't have data yourself, you can actually then mine the literature, generate hypotheses fairly easily, and then use that to further, you know, do other work, you know, to get, you know, a grounding point where you can move on to other you know, discoveries.
One thing I should have done when I did my intro is actually define bioinformatics today. What do you guys think bioinformatics means today, and how has it evolved? And Katie, you can go first.
Well, if I think of bioinformatics, it's really all the way from getting your raw data, doing the pre-processing and QC, which is really an essential part of the data. It's probably the most important part of it, if not the most interesting part. And then, you know, downstream, what is your research question? And that will drive the type of analytical tools that you want to use. And, you know, various visualization tools to get a look at the data first. And then the downstream statistical tools, which can include simple class comparisons or biomarker discovery with machine learning, functional analysis, pathway analysis. So it's really quite broad, and I think that keeps it interesting. And, you know, it really just depends on the research question and the disease you're looking at.
And Afshin, how would you define bioinformatics, and how has it changed over the years?
Yeah, I mean, I think I would probably define it similarly to Katie. I mean, a lot of it is, yeah, how you do all the processing of the actual bigger data, and the QC is obviously the most important thing. So if your data doesn't have good quality, it's garbage in, garbage out, right? So, but yeah, so bioinformatics, I would define like the use of big data, basically. That's it, and making the data understandable. Simply put, you know, understandable for a biologist to say, all right, now guide, you know, let the biologist guide to generate hypothesis downstream, doing the fun, you know, higher order analysis on it. So in a nutshell, I think it's the interplay of the big data sets to actually make it understandable for the human to look at, because otherwise, you know, you get this massive data and people don't understand it, or they might cherry pick again back to their favorite genes or factors of interest.
So things have certainly evolved since 1995. And to me, like bioinformatics back then was DNA microarrays. Um, where do you see bioinformatics applied most often in your research? And how about Afshin, you start?
Sure. Yeah. I mean, that's true. Yeah. Back when we started, yeah, it was just DNA and RNA, but now bioinformatics includes everything: proteomics, lipidomics, proteins, lipids, metabolites. You know, everything's encompassed into that. So for me, in my research, it really is an essential tool, because it's a, you know, it's an easy way to generate a hypothesis when you don't have to do the experiments yourself, because then there's a lot, I said, a lot of publicly available databases, so I could quickly mine the data, figure out if my hypothesis would be true, and then you could apply for grants or funding to figure out if that would further validate these results. But so bioinformatics is and another part is when you do run an experiment and you run the bioinformatics, it gives you truly a quick way to figure out the key pathways that are being regulated, and let's say within the systems you're looking at, experimental versus your control. So that's for me, it's pretty much the central thing where these days I might use more transcriptomics, like look at the RNA, just because it's one of the cheaper omics platforms and tools out there. But when you do have funds and research to do them all, like you want to incorporate everything from the proteomics to the metabolomics and so on, to then you get the complete picture of what's happening in the research. But yeah, in my research I use it 100% of the time. And then of course you would need wet lab confirmation and wet lab experiments to complement the bioinformatics too. But you need both basically.
And Katie, how about you? Where do you see it being used most often?
Well, for me, I am now fully in protein microarrays. So that's what I deal with every day. In the past, I worked with different data sets. But, so my main interest and focus now is the protein microarray data. So to get the immunoprofile of an individual. And what links onto that is data integration with other omics types, which is also a very interesting field, and to see how can we merge those to get more out of the data than in any one of those individually.
So you gave us a training last summer, and I remember you talked about your genomics background a little bit. What was it like moving from genomics to proteomics? And what would you say were some of the challenges and differences between working with proteins and DNA?
Well, I think because in my PhD I used gene expression microarrays, I could apply a lot of the things that I learned there. It's also, I think if I moved from like gene expression microarrays to mass spectrometry proteomics, it would have been more challenging, as there's quite different fields. And I actually remember in my PhD, which was actually in a proteomics lab, that I did my genomics project. I was grateful that I didn't have to analyze the mass spec data, and then I had my gene expression data. So I think a lot of what I learned from quality perspective and processing, I learned on the gene expression platforms that I could apply to the protein data. Obviously the chemistry is different, the data structure looks different. But a lot of it is transferable. So I think that step wasn't too big. Once I understood and appreciated how the platform worked, how the chemistry worked, and all the intricacies of all that, then kind of got a handle on it.
Afshin, do you have anything you wanted to add?
No, I mean I think that was well put. I mean I think the main thing is a lot of these pipelines that are being built are not by biologists, but they're by computer scientists or mathematicians or physicists that come along that would build these pipelines. But so after you get over the initial processing of the data that Katie mentioned, a lot of the analysis always seems the same between transcriptomics, proteomics, metabolomics, because now you just have the numbers, and now you have the numbers. Now you can actually run the same statistics after that. So I think it's really applicable once you get to that stage. Of course, the pre-processing and the platform is a huge, endeavor to try to understand, since that's very different. But that's the hurdle that I think Katie did very well, right? And other folks that might have tackled when they come along. So you would definitely need like a proteomics expert to do all the pre-processing, because that's going to be very different from transcriptomics, as Katie mentioned. But once you get to the point where you actually have the data analyzed and the higher order data processing, then it becomes very similar to the pipelines and things you could run, with analysis.
So next gen sequencing has appeared to have supplanted DNA microarrays. So I was wondering if people still run DNA microarrays, and if so, for what purposes? Does DNA microarrays still have a home, so to speak? And either one of you can answer that first if you'd like. Katie?
Yeah, I mean, I haven't analyzed DNA microarray data in a long time. I think RNA has largely taken over. But I think for clinical applications, gene expression microarrays can still be used for kind of more of a targeted high throughput setting. But maybe Ashton has something to add to that.
Yeah, man, I'm sort of in the same boat. I haven't done it in a long time. I think most folks have doing the RNA or RNA-seq. I think it's a yeah. I think it's good if it's a, probably if you don't have much funds to run, let's say the sequencing is probably a quick and easy way to get a targeted approach and some specific thing you're looking at in the clinic. But typically now most people don't really tackle the DNA microarrays as much as I know, because now they go because the sequencing has become so cheap. It's almost the same cost as to run the sequencing, and you get more information out of it. But that said, I mean, you know, some people might want to run the targeted approach more, and then the DNA microarrays might be something they might fall back on.
All right, well, let's talk more about protein microarrays because they're still very valid. Katie, what do you think are some pros and cons of protein microarrays?
Well, I think if the goal is to do immunoprofiling or, get the antibody repertoire like we do, the main advantage of protein microarrays compared to something like mass spectrometry is that, with mass spec, it's really hard to profile, to distinguish between antibody sequences because they're so similar. And that's where your protein microarrays really have an advantage. I think also from a practical perspective, just the equipment that you need. A scanner is much more common lab equipment than a mass spec, and it's relatively simple tools, washing and scanning the arrays. So that makes it more accessible as well. Data analysis also, if there's an existing pipeline, it's pretty easy to run and not super computationally intensive as well.
How about you, Afshin? Do you get involved with a lot of protein microarrays? What do you think the value is?
I haven't, but I know people who haven't. I think the value is actually running proteomics is a massive undertaking and very expensive if you run especially untargeted. And the protein microarrays, I think, is a nice, very good substitute for people to run it. And you don't need to be an expert in proteomics to actually analyze that data. So I think right now I think it's a good technology and a substitute for proteomics, like running the mass spec and doing all that massive data they need to be around. So I think proteomics is where transcriptomic was like 12 years ago, 10 years ago, or 15 years ago. That's why we need the protein microarrays now, because I think that's the technology that will be very beneficial for everyone, and affordable, I should say, too.
So let's talk a little bit about setting up an experiment. You'd mentioned cost, but Katie, what are some of the things that go into designing a study for the protein microarrays?
Well, I think in terms of the protein microarray data, study design is very important, and specifically considering sample size. So the data structure is quite different to something like gene expression data, where, often if we've got two classes in gene expression data, you'll see like consistent upregulation in one and consistent downregulation in another, and it's like, really clean blocks on your heat map. Whereas with antibody data, it's a lot more kind of stochastic, the patterns. And that means you often need slightly larger sample sizes to get to the power that you need. So, as part of what we do, we like to speak to clients when they design the study, so we can help them set up a properly powered experiment. And there you want to consider, what's the variation in your data set across your population, what's an acceptable FDR to the question you're asking, what kind of effect sizes are you happy with, and then you can really decide, do you have enough samples? If you really don't, can you consider a pooling strategy? So those are kind of some of the considerations that we like to think about before we start a study.
Everybody probably knows this. I just want to be clear. FDR stands for false discovery rate. The first time I saw it in a scientific publication, I was thinking President Roosevelt, but I knew that wasn't what it referred to, but I had to look it up. FDR is false discovery rate. So what went into developing the Syngenix platform, Katie? What were some things that you initially tackled?
So I think, the way this pipeline evolved was really kind of carefully considering the platform and the technology and the chemistry, and what was required to get the highest quality data. So on our platform, we've got 4 replicates per antigen. And using that, we can ensure we get high reproducibility for each antigen. And then, after that, we consider what kind of normalization we need. So, just considering the various steps that you normally would in a bioinformatics pipeline. So from QC to normalization, to feature filtering, which is also a very important step. And I think all of those stemmed from really understanding the specific data structure of our platform and evolving from there. So once you get to the high quality, clean, filtered data set, then I'm happy. Then you can, apply many different techniques to analyze the downstream data, like Afshan mentioned earlier. We use linear modeling, we use random forest, and these are types of techniques that you can apply to many omics data sets. So yeah, I think we're constantly evolving and improving our pipeline, and also making it more user-friendly by, building apps that anyone can use to analyze their data. So yeah, I think it's a continuous process.
So Afshan, I know you said you work with a lot of transcriptomics. Is there a theme, like a particular application that people are most interested in with their research, such as biomarker discovery or target validation or target identification, maybe?
Yeah, I mean, depends on who your funding agency is, right? And then what grants you have, and what your focus is. Because some people obviously want to look at especially clinical things. If you could come up with a nice biomarker, right? So doing a biomarker targeted study might be great, because then you could find new biomarkers, let's say for cancer or for other Parkinson's or Alzheimer's or other diseases, right? So that's one aspect of it. But then also, like, it depends. Some people want to do more of the basic science. So it depends on, let's say, if you have money from, let's say, National Science Foundation, NSF, they might be more basic science, that they want to know what's the biology really changing. So that means sometimes when you run your massive omics pipelines and tools, then you would come up with like pathways and really understand the basic biology of what's changing within the, when you use your omics data. But then, I think clinically, I think it's biomarkers, but also then can you use this data to potentially come up with targets for therapeutics or countermeasures, depending, let's say in space research, you want to come up with countermeasures to prevent the damage being done. So, for example, I have collaborators who we use like AI machine learning techniques to, once we run our bioinformatic tools, we come up with some candidates for biomarkers, and then also use machine learning tools like knowledge graphs to mine the omics data to come up with potential like therapeutics that could target your specific, experimental setup for your control, like cancer, for example. So, I mean, the majority of work I do is probably more biomarker and countermeasure therapeutics, but then the byproduct of the analysis you do with the omics, whether it's the protein micro or the proteomics or the transcriptomics, you also understand the basic biology and the process of it too. So
You'd mentioned AI, and I think you mentioned one model. How does AI factor into bioinformatics? What types of tools do you use? When do you apply it? What's required to use it?
So yeah, so AI and machine learning techniques are basically up to the creativity of the user. But I mean, that's the thing, there's no standard like pipeline people use. People are coming up with different things to tools. So with one collaborator, for example, we created a machine learning technique to, one type of research I do is look at microRNAs, which is very small RNA that circulate in your body and found in many tissues, and they target genes, basically, hundreds of genes, these really small microRNAs. And one aspect of this is a biomarker, so they circulate in your body. But we use a machine learning technique, looking at microRNA sequence data, and then seeing how, for example, the key microRNAs that might pop out, how do some small molecule drugs that are already approved by like the FDA in the databases that are out there, how can they target the microRNA? So he's developed a machine learning tool to actually do that. Papers under review right now. But that's something like, for example, one example of using AI or machine learning tools to say, use the omics data, and then you plug in that into the machine learning tool to predict drugs that could potentially look at the chemical similarity between the drugs and the sequence similarity of the key factors developed from your omics tools, to say now you got potential already approved drugs that people haven't thought of that you can repurpose for what you're looking at. And then you could also use that for transcriptomic data or proteomics data. You could just plug and play, take out microRNA and put in your proteomics data, use that machine learning technique to now look at that. And there's other, examples too, like knowledge graphs a lot of people are developing. And knowledge graphs are basically, in a nutshell, it's using AI to mine the thousands and millions of data that's out there in databases, and then figure out from this big hairball of network of things happening, whittle that down to basically the key components that are, you say the question is, if you want to come up with a drug, you put in your transcriptomic data or proteomic data in there, and then it whittle it down to maybe you got 20 candidates to it. I mean, oversimplifying what's involved, but basically it's looking at thousands and millions of databases, and then something we can't do on our own. And then what usually what happens is usually knowledge graphs and AI will pull out key pathways, key targets, key things that we'll never be able to do manually. So that's in a very basic way. That's the AI and ML tools that I incorporate. But there's also lot of other things that people do that I don't collaborate. I'm not even aware of. But there's a lot of different things people do with AI, ML, and omics.
Katie, you want to add anything about AI and bioinformatics and perhaps your experiences?
Yeah, I mean, I think the machine learning application is a very useful one. I think recently as well, with kind of GPT-3 input, I think there's pretty much a new app every day. I think a lot of them will become useful for our day-to-day bioinformatics tasks, from debugging code to enhancing user experience in our analytical platforms to helping us with image analysis. So I think the possibilities are endless and quite entertaining, and I think it's something it's going to change bioinformatics quite a bit, I think.
So leading in to that is, how is the data handled? Like when you get the data and you start processing, what are some steps involved, and what does the output look like?
So I think in the majority of kind of bioinformatics pipelines or workflows, regardless of what language you use to code the pipeline, there's always going to be a QC step to assess the quality of the data, some form of pre-processing. You may be filtering, reads if it's RNA-seq, or spots if it's a microarray, failing certain samples if they're not high enough quality, and perhaps rerunning them. And then once you're happy with that raw data, there'll normally be normalization step of some sort to make sure you can compare between samples, and then also a feature filtering or antigen filtering step. So those are common to, I think, most omics data types. And then after that, you'll start with your exploratory analysis. So you'll look at different visualization techniques. We often use heat maps because you can see all antigens, all samples at the same time. So, in addition to the pipeline, you have your eye becomes highly trained to really spotting anomalous patterns and also interesting patterns in the data that you may miss if you just push it through a pipeline and not constantly look at what it is that you're working with. Also ordination techniques like t-SNE, and then the downstream more common things like just using a violin plot and a volcano plot to look at hits that might be of interest for you to follow up with orthogonal platforms. And then we also do functional analysis. We do random forests. We do unsupervised clustering for subtyping. So yeah, it's a nice mix of different tools that we have.
Afshin, did you want to add anything?
No, yes, all the examples Katie mentioned, yeah, pretty much it. Yeah, usually once you get to the higher order analysis, like you run your standard toolbox, but then there's obviously a lot of packages now coming out more and more from people developing newer ways to analyze that. Once the processing is pretty standardized, at least for transcriptomics, there are pretty accepted pipelines that you'd use. You don't want to deviate from that. You want to make sure your data is comparable to everyone else, unless you're doing some specific analysis that's different, answering different questions. But once you get to the higher order analysis, the standard stuff that Katie mentions, you run all those because that's the basic things you want to do. But then there's now a bunch of other packages that you could find in R programming and Python that come up, and people write new ones every day. For example, creating ways to pull out data that we might not be able to do on our own. But it is basically, if someone does a Google search or go on Bioconductor, which is one of the R package resources, you'll find hundreds of packages there and more popping up every day. So there's lots of different ways to do this, and people develop more on their own too, a lot of times.
So Afshin, you hit on two things in the last couple of minutes. One was on standards, and then you just mentioned learning. Are there industry standards for processing the data, and what do you think is needed in the future?
Yeah, I mean, I don't know if there's a accepted industry standard, but I think the community accepts it. So, for like transcriptomic data, it's been around for such a long time. RNA-seq data, there are pipelines that have been pretty much well established that most people use the same pipeline almost. They might have some standard deviations from each other, like in the parameters they might use when they process the data, for example. The cutoffs they might use for the pre-processing, but usually that one's it's pretty much accepted by the bioinformaticians. Like, yeah, you would run this pipeline, you might change some of your parameters here and there, but that's overall it's not going to deviate from too many people independently doing it. Yeah, I mean, and then once you get to the higher order analysis, then there are standards there, for example, for your statistical cutoffs that people do use, but the statistical cutoffs could sometimes be, depending on, let's say, your data set you have, could be too stringent sometimes. So let's say for, you mentioned FDR as a false discovery rate that people first use for omics data. You might want to lower that sometimes just to see, say if the data is noisy and you want to pull out information. Sometimes you lower that and just mention it and say, look, we lower the statistical stringency because this data set, we want to try to see if something's important, and then we'll validate in the lab, and then you might have more false positives. So that part of it, there are standards accepted, and then you can deviate from that if you justify it accordingly, not just do it from your whim. But obviously when you do things scientifically, you got to justify everything you do. But I think in the proteomics world, it's a little different. Different than the omics. When I talk to my proteomics colleagues, I'm sure Katie might agree. It still seems like there's the Wild West a little bit where, there's depending on which proteomics person you talk to, they have a different way of analyzing the mass spec data, which then you've got to find the one you trust. But it sounds like, like the protein microarray, like microarray kind of things might standardize that a little easier, because when microarrays first started, DNA microarrays and started, people figured out how to standardize that analysis fairly quickly since it was easier to do than the same mass spec.
And Katie, what do you think? And what are some advantages working with Syngenics and our technology?
Yeah, I agree with Afshin, and also just on the protein microarray space. There you really like also considering the specific platform for the pipeline in terms of the QC and the processing. So that might vary a little bit depending on the the company that you're working with. But for the rest, I think the rest is pretty easy to standardize. I think from the Syngenics perspective, what we really try to do to limit false positives in the data is to mirror as closely in vitro on a chip. So, the correct slide that you're showing now is to make sure that the proteins are properly folded in their native state and properly oriented on the array, because that's really where we get biologically relevant signal from. So often in nature, you'll get antibodies binding to discontinuous epitopes, and to get that signal, you really want a properly folded protein. Once the protein's unfolded, or you work with a peptide, then you're going to start, exposing those hydrophobic surfaces and start inviting non-specific binding. And I think that's something that's very important to consider when you choose your platform. That's along with making sure there are a suitable number of replicates per antigen. So on our platform, we've got 4 reps per antigen, and that really ensures that we can have low CVs and high reproducibility. So I think those two are main, those two things aren't negotiable. They're very important to make sure that you're getting biologically relevant signal. Otherwise, you'll get signal, but you won't really know if it is something that'll validate.
So we have a a question from one of our guests. How do you deal with misclassified data where up to 30% of your disease group classification is wrong, but you do not know which are wrongly classified?
Well, I hope my client doesn't supply me misclassified data. I'm not sure in which context you mean, the misclassified data. Are we able to distinguish which are misclassified, or so I can give you maybe one example. So we often see, if we've got paired samples of maybe pre and post treatment, we sometimes see we can pick up labeling, sample mislabeling errors, because the individual's profile stays relatively constant over time, and our technology is highly reproducible. We can see a high correlation between those samples, and if it matches up with a different individual, we know there was probably a sample mix-up. But yeah, I guess it depends what you're looking for exactly.
Afshin, did you have any
Yeah,
thoughts?
I mean that's the tough part. It's he misclassified. Yeah, I mean, I guess it depends on what the question say. If it's actually the samples itself, that's a tough one to actually I mean, a lot of times you run, let's say, as Katie mentioned, you run your PCA or Tisney plots, you see how the samples cluster, and if you see like obvious samples consistently clustering with like your experiment, which should be your control, then it could be biology, but maybe someone misclassified that and mislabeled that early on. So that could be one way of looking at it, but then you still don't fully be able to justify that. Oh, an aneuploid. Oh, okay.
Yeah, this is from Ray. Ray is a PI. I can't remember what institute Ray is at in England, and he just started. He just opened up a a mass spec core, a proteomics core. So
Yes.
he is an expert in mass spec.
Yeah, I see what you're saying in the chat now. You're saying that, yeah, so that's different misclassification. So that I think there's, I mean, Katie might want to expand on this. At least in the I'm not the mass spec expert, but I know, let's say in the omics data, yeah, you get sometimes misclassification there. So that's I think just the database itself has to be the databases have to be continuously updated, which they don't. Usually you have to wait for that. The other part is I think this the machine learning AI tools that are out there now, like the knowledge graphs I mentioned, sometimes they could pick up on these things because they're tackling millions and millions of databases that, let's say, if you use your standard pipelines, you don't. And there you can actually pick up some of these misclassifications using the knowledge graph and machine learning tools that are out there. But then you have to collaborate with people who know how to do that. But I've been able to actually figure sometimes, yeah, when you run your standard pipelines and you see this misclassification based on what you're seeing here, those knowledge graph and AI tools are actually a good method to pick that up.
Yeah, so I think essentially what you're mentioning is that your data is going to be more noisy, so there's going to be more error in your data. So I think you'll need probably a larger sample size to account for that, and then validate it to see if what comes out again.
Thank you.
And I was going to say also, there's also sometimes, with the single cell RNA-seq technology, for example, they've been able to go back to the bulk RNA-seq and then be able to translate their technology that's there to be applied to, like, classifying, let's say, cell types that you previously couldn't have in the bulk RNA-seq. So there's tools out there now, and I think you put in the chat, is there a way for machine learning allow for such people are starting to develop that. I don't work with any of those folks, but I've seen papers on that, so I could always find papers and send and I talk to my AI machine learning folks, because I know they've looked into things like this. It's not an easy question to tackle, though.
Well, that actually leads into my last question, which was, and I know Ray, so if you have references that either of you think would help him, you can send them to me and I'll make sure he gets them. The last question was basically, where do people go to learn more? Do you have any resources or folks that you think people that are interested should go and look into?
Well, I think for learning bioinformatics, there are a lot of ways to go about it. I think having a mentor is really great. I think it, stretches you and teaches you new things. For instance, in my postdoc, I had a really good mentor And that, that led me to really build robust pipelines in a way that maybe I wouldn't have otherwise, using specific, workflow languages. But I think that being said, short courses in bioinformatics are a great way to go. You can definitely teach yourself. A degree is great, but I don't think it's essential. And also, bioinformaticians are quite a friendly bunch in general. Usually you can just ask, very good on email. So, I've always had good experiences with it. If I ask people about how they do their analysis or how does this package work, you'll get good feedback and probably good collaboration and learn a lot from that as well. So I think various sources and just patience and time.
Yeah, I think I agree with Katie. The bioinformatician community is very friendly and usually they're more than happy. I think that the type of, like, they want to share their knowledge with everyone. So they're more like the open science. They've embraced open science and they say, hey, let's help everyone do. So I think to expand on that, there's also these I'm sure people have done this and they Google, how do I do this in R? How do I do this in Python to do this? There's these nice communities out there like BioStar and Stack Overflow that I use all the time. If I'm stuck, and like I go in there, put it in the forum, and then within an hour they got like 10 responses, and this is how you actually do it. So then, and then if you have errors, you just put your script again, say, hey, I got this error, and then within within a good day or two, you might have solved your problem, and the community comes in and helps you nicely. Other than the fact that you can approach and say people too, and that's another way to do it. So yeah, I think the easiest way is talking to people like Katie said, but then using these communities that are out there now online, like Stack Overflow, BioStar, there's other ones too, that really is a good forum that bioinformaticians constantly are on there using, and some people just want to help. So yeah, the network, you grow your network, that's the way to do it. And then have people talk with you. And oddly enough though, Chat GPT sometimes helps you coding. I don't know if Katie's used that before. One of my colleagues once said, if you have an error, put it in there. So I've done that, put in my script and say I have this error. After 10 iterations, I actually solved one problem once. Then I have to use it more, but that's another one that surprisingly worked well, is to solve a bug in my code and just figure out how I can do something. So using AI to your advantage that way might be something that you want to explore too. So.
Oh, good to know. Well, we're just about out of time. I really appreciate Katie and Afshin for joining us today. That means a lot to me personally, and hopefully everybody learned a lot today and is more excited about bioinformatics and protein microarrays. And this is something that I wanted to continue doing, these webinars. So we have another one scheduled for next month, and this will be more of a presentation style with Janique Paper, who is systems immunologist on Syngenics staff. And I'm pretty excited about this one because she's an expert in a lot of things, but especially B cells. And throughout my career, I was a developmental neurobiologist. I went to adjunct work for Seahorse Bioscience, a lot of knowledge with cellular respiration. And I have a good feeling for things like T cell biology, but in my experiences, whenever I would ask people about B cells, they were like, "Oh, well, I'm not an expert in that," or "That's kind of complicated." So Jan's going to be talking about B cells and how they relate to disease and how they can be used. I don't know the details yet. As we put that together, I will send you invitations to the meeting and more details on the topic. Some things that I'm particularly interested in are things like headless CAR and bispecific antibodies. And these are things that B cells could potentially play a role in. And hopefully Jan will talk about those, but she'll definitely teach you all about B cells, B cell physiology, and antibodies. So be on the lookout for that. If you want to mark your calendars, that'll be October 12th. I haven't set a time yet, but that's coming up. Let's see, a couple more. I did protein deconvolution analysis, and I'm not sure how to validate the cells I identified. Is there a suggestion to how to validate these cells? And this is from Javan at NIH. Any thoughts before we sign off?
That might be a Katie question, so I don't deal with proteins that much, but that's a tough one, I think.
Yeah, that's a tough one. I think I'll need a little bit more context. I'm not sure it's quite up my alley, but happy to chat about it more afterwards.
Yeah, I should get a transcript of the question. So Javan, I'll hold on to this one and I'll talk to Katie and see if we can't come up with an answer or some references for you. Otherwise, thank you everybody for attending. Hope you all have a fantastic weekend. And for those of us in the United States, have a happy Labor Day. Otherwise, stay tuned and thanks again. Take care, everybody.
Thanks,
Bye.
Kevin. Cheers. Bye, Ashley. Cheers.
Bye.